screen sharing by any chance? So thank you for that, Lawrence. As Lauren said, my name is Gabrielle, and I am a fourth-year medical biophysics student at Western University. Today, I'm going to be presenting my research, which looks at using image analysis techniques in Fiji and MATLAB to extract quantitative characteristics from microvasculature networks. So during this presentation, I'm going to be focusing on a couple of main topics. I'm going to start with why study the microvasculature at all. Then I'm going to move on to giving a look at the general process I use just to provide a bigger picture to keep in mind as we go through the presentation. Then I'll go into what I did specifically using Fiji and MATLAB. And at the end, I'll provide a summary, a little glimpse at some of the results I've gotten so far. And I'll also I'll take any questions at that time. So why study the microvasculature? The microvasculature is responsible for the direct interactions between tissues and the cardiovascular system. And most of the time, it's very crucial in health to deliver essential nutrients and oxygen to different tissues with different metabolic needs. It also has roles in wound healing and mediating the immune response. So needless to say, it's pretty important. And because it is so important though, the failure of microvascular networks to function efficiently can have very serious and even life-threatening consequences at times. These failures or abnormalities may actually stem from the network itself, or it can come from different areas in the body and be a symptom or a result of other diseases, just because of how highly interconnected it is. And because of this fact, it's actually very commonly the latter as well. The figures on this slide show the top 8 causes of death in the United States, which are the same in Canada, but in slightly rearranged order. So as you can tell here, the roles of the microvasculature in these diseases are very abundant, and there's a wide variety of different roles that they hold. In some cases, like in accidents and injuries, the microvascular networks respond in ways that you want them to respond in order for you to heal. But in other cases, it actually helps the disease progress. So in cancer, the pathological angiogenesis and capillary hyperpermeability are both things that actually help tumor metastasis occur. So now that we know that there's like roles in health and disease, this is where quantitative characteristics of networks come into play. The analysis of networks quantitatively can be a very powerful tool to provide insights of what the differences are in networks between health and disease. This figure shows some of the most common quantitative characteristics that are typically extracted. So the number of branch points or bifurcations, the vessel length, the vessel segments, the tortuosity or how twisted a vessel might be, the vessel area fraction in a given field of view, and the diameter of different vessels. All of these metrics provide different insights to how the network might be working. For example, with bifurcations, they actually let you know how interconnected a network may be. And by knowing this, you can sometimes estimate what might happen to a network as a whole if an occlusion were to occur and how much damage there might be in that case. So you can also use these metrics comparatively over time or by analyzing different states of a disease. So maybe at one point in health and then one point in disease. And this can give you insights of whether or not maybe the network remodels or if vessel density might reduce and what that might mean and what implications those might have on the networks and the entirety of the system. In my research, these are actually the networks, the statistics like we aim to get. So in my research, I used 3 main applications, 2 of which most of you are probably familiar with. I used Fiji or ImageJ, Procreate, and MATLAB. Fiji is where majority of my image analysis was done. In MATLAB, I did minimal image analysis and a lot of data visualization. And Procreate is the one that most of you may not be familiar with. So in Procreate, it's essentially a raster graphics editor. So it's mainly used for digital art and design. And it is a very common iOS app. It's essentially a very precise drawing tool, and we'll see what I used it for in the slides to come. So this was my overall process, really. This is where most of my work was done and most of my time was spent. Essentially what I did was we had intravital microscopy videos of a microvascular network. And from that network, I would draw the vessels that had visible flow and had less ambiguous edges. The reason why you wanted less ambiguous edges too, like you want a defined edge, is because a lack of clarity also will lead to a lack of quality in your results when you're getting a quantitative measure. So blurred edges or things that weren't clear aren't very good candidates for analysis. So what I did is I would take these videos, I would process them in Fiji into frames, and then I would take a frame, put it into Procreate, and I would draw based on observation what I saw. So this image is just an example of what I drew. The different colors in the image are representative of different vessels or different layers. This is just to avoid confusion with bifurcations. And this part of the process can be done in Fiji, it absolutely can. It's just, for me, when I was using Procreate, I thought it provided me a lot more, I guess, control because of the fact that I had a pen versus a mouse. And it was also very efficient to use because of that control. Now, when I had this image, this annotated image of the network, this provides me with center lines. So center lines are not representative of the entire network. This is because it lacks the thickness of the vessels. It's not completely representative of the vessels that way, but you can still get a lot of information from them. You're able to get the number of bifurcations, vessel segment lengths, and even the number of vessel segments from Fiji. I also measured the vessel diameter in Fiji. However, I did not get this from the center line alone. I would have had to make markings for the diameter across the width of the vessels and then measure that. So because it's not completely representative of the network, I went back to Procreate and I annotated networks of the entire vessel. So this is representative now of the diameter, and you can get things like area from this. So I would binarize it in Fiji, and then I would put the binary image into MATLAB to get the vessel area. So how much of the white in here is taking up the entire field of view would give us the vessel area fraction. So using Fiji specifically, what I used in it was actually pretty simple, and I didn't use very much. The main tools I had were the segmented line tool, the multi-point tool, and the magnification, just for clarity, because every day it seems my vision gets a little bit worse. But so you use the multi-point tool to count things like bifurcations. So you just plot them there, and then you can measure at the end using the command M function. This here isn't a bifurcation. That was a mistake I made, but this is just an example too. This is for the segmented line tool, you're able to use it to measure the segments of the vessels. So you kind of like plot it like it's a line of best fit, and then you measure it and it tells you how long the vessel is. As for binarizing the image, I would go into process, make the image binary, and make the image binary by going to the binary function. It's very simple. Now, with this binary function, MATLAB has a function to actually just tell you the area of the white parts of the image. So BW area does that. That's what I used, and it was very simple, a very quick process. You can also do this by thresholding, but I don't think that there's a need to, just because this is easier. And obviously MATLAB is also a very powerful image analysis and data visualization tool. I have only scraped the surface of what it can do with this project so far. And I think that's something that can really be looked into further. So beyond what I've done with it, I'd probably be interested in further seeing how to automate this entire process of edge detection and then the extraction of the characteristics from the vessels in MATLAB, because that'd be pretty cool to look into. But with what I do have so far, I do have some results. So this is indicative of the bifurcations across different experiments. Each experiment is one field of view of a microvascular network, each experiment containing 2 videos, one at time equals 0 hours and one at time equals 3 hours. So this was imaged in rats, and at 0 hours, these rats were healthy, but then by the 3-hour mark, at some point along the way, they were given sepsis. So across experiments, bifurcation count tended to decrease, as you can see. with the exception of experiment number 9. In experiment number 9, the bifurcation count increased, so the network became more interconnected for some reason. And this is obviously like abnormal and anomaly compared to everything else. And it may have happened potentially because of the fact that the network might have just responded that way. There could have been human error when I was measuring things, or it could just be that the field of view shifted as well. And there were a different segment of like the network was visible at that point. But to figure out why it happened would be pretty cool to continue to investigate as well. That brings me to the end of my presentation. So in summary, studying the microvasculature and getting quantitative characteristics from networks can tell us really important information about the mechanisms of the system in disease and in health. I personally used Fiji and MATLAB to get my quantitative measurements. So I used Fiji for measurements such as length and counts, and MATLAB for measurements of vessel area. And thank you all for taking the time to listen. I've included my email if anyone has any questions or further points of discussion, but I'll also take any questions right now.
Thank you, Gabrielle.
No problem.
Prof. Frisbee has a question. Gabrielle?
Actually, I have several questions. But nice talk, very interesting. I think it sounds like you're laying a really good groundwork for moving forward with a lot of this. I have them all written out in the chat, but that just seems awkward. What does interconnected mean? Let's start with that.
So interconnected in the sense was just how many vessels are connected to one another in the network, the segments, how connected
aren't they all connected to something?
Well, it wasn't necessarily like an endpoint, but it just meant how many branched off of one another. So had the same spout of flow, I believe.
So are you talking about microvessel density, the numbers of parallel pathways? Like what are you referring to? Like I'm not sure what interconnect, that's what I'm struggling with. Like we can have like a cancerous tumor, which is highly interconnected, but it loses its fractal branching pattern and it's much more chaotic and other problems. But I'm just trying to get a handle on what interconnected means in this regard.
Okay, so
I know what I think it means. I'm just curious what you think it means.
Okay, can I show I'm going to share my screen again just to show my image. I might need like a visual aid to explain.
By the way, how much time do we have for questions? I don't want to go too long or anything.
We have all day, Prof.
Okay, that's a little harsh, but.
So for me, when I think of how interconnected it is, when it branches off, these continue to branch off to other vessels. So I took it in that regard.
So are you talking, if I use the word Strahler ordering system, do you know what that means?
I do not.
Okay, it's just a nomenclature that people use to break up microvascular networks based on size. So when you say interconnected, you mean how many branch points or how many bifurcations do you get down to the capillary level and then back out again on the venular side?
Yeah, so within that one level pretty much is how connected it is, because yes, everything is connected at the end of the day. I wasn't too clear with that, sorry.
That's fine. That was easy. Okay, so you mentioned when you put up your image from your prep. If you could skip to that.
This one?
There you go. First of all, what is that?
This image?
Yeah. What tissue is that?
I believe it's the EDL muscle of rats.
Okay. So you said vessels with visible flow.
Mm-hmm.
So you saying you had vessels in there without visible flow. What I'm assuming that's what that means.
Yeah. So if I couldn't tell that there was movement within the vessel, like it wasn't clear to me that there was something moving, I wouldn't make a mark. So
what diameters are you down to? Because arterioles are going to be, consistently perfused. But when you get down, certainly as you start moving into the capillaries, then you're going to start to see off again, on and off flow, or you might actually get plasma flow without the presence of red cell perfusion?
Okay, so some of them had visually like stopped flow. There was nothing moving through them at times, or the blood cells were like stuck.
So you can see red cells just stationary in the vasculature?
Some of them, yes.
Okay.
This one, I don't think there is.
Okay.
I can't tell from this far away. But
that's actually going. Okay, I couldn't tell at all. Okay.
Yeah, it's moving.
Oh, there it is. Yeah, okay.
Yeah. So if there was something that, like this right here, you don't really see. From here, I can't see any movement, but I think I was able to when I zoomed in. So I just wouldn't record something that I just didn't see movement through.
Okay. What did you, how did you? And did you classify divergent versus convergent flow at all? Like you use red and blue, so that's gonna make everybody think that means something.
Oh, I didn't. So the red and blue were just to separate whether or not they were separate vessels, like when vessels overlapped, but they weren't connected. So I wasn't going to confuse them for bifurcations.
Can you go to your data?
I can.
Okay. 11 out of 12 of these go down, as you mentioned.
Mm-hmm.
1 out of 12 is an outlier and goes up. And yes, there's 1,000 reasons why that could happen. Do you think, in your opinion, the fact that the number of bifurcations that you can pick up based on a hemodynamic observation is due to a reduction in prep quality over that 3-hour period? Or do you think there's something else going on?
Sorry, so you're asking if the reason this went up could be because the prep wasn't as good?
Well, I would look at this and say this is a time control experiment. Mm-hmm. So, you started at hour 0. So in model 1, you've got 32 bifurcations that you can pick up based on red cell movement. Mm-hmm. Right?
Yeah.
And then you come back 3 hours later and you're down to 24. Mm-hmm. That suggests to me that is probably the slow decay in the quality of the preparation and the quality of the animal on the table. You could also get things like a progressive inflammation over the time of the experiment that will then, then you got white cells, they start showing up, they block things up, and your bifurcation shuts down. Okay. So I think there's that actually, you could have a lovely time control experiment here. I think that may be what this is telling you. And that one outlier, it's biology. We get outliers. So, but I think that's actually, when I look at that, that's how I would interpret it. Is this is the foundation of what you do. So if you do a subsequent experiment that has a 3-hour intervention associated with it, or a 3-hour disease state, like do this in diabetic animals, and you may see that delta from 0 to 3 becomes much higher because they just degrade faster. So it can be helpful in interpreting your data if you know that.
Okay.
So I see that Keith also has a question, so I'll shut up. But that was a really nice talk, and I look forward to hearing more about this as it moves forward.
Thank you for your question. So perfect. I'm just going to read your question right now, Keith. Sorry. Should I read it out loud or?
Please do. Yes, Gabrielle.
Or you can just ask it, Keith, if you want.
Yeah, I'll unmute myself really quickly. It was just a great presentation, Gabby, and I just wanted to ask, you're using the network as a unit of measurement, and are you just defining your network based on what you can see in your IVVM field of view?
Yes, the measurements are all taken in pixels for the most part, because I didn't know the magnification of the field of view. I know in Fiji, you are able to adjust it to represent the actual measurements if you know the magnification, but I didn't.
Oh, I see. Okay. And another question I had, and I think Dr. Frisbee's great questions touched on this a little bit, which is he mentioned convergent versus diverging flow. What the first question is, do you know if your field of view and the measurements you're taking is always in the same part of the microcirculatory network that you're looking at? And can you tell if they're coming away from a terminal arteriole versus going into a collecting venule? Because depending on where you're looking at, there might be some differences in bifurcations there too.
Yeah, so for the fields of view, they were in the same area in experiments. Okay. Some of them did slightly shift over time. I think that was just the nature of the setup. But as for the convergent and divergent flow, I could tell which direction they were coming from. I just didn't make note of it in my research so far. So that is something I should probably consider doing. But yeah.
Okay. Yeah. I'll second on to Dr. Frisbee that I really look forward to see how this goes. This is really cool.
Thank you.
Can I comment on a couple of things, please, Gabrielle?
Yes,
sir. So firstly, you have to take on board all comments that have been made in terms of scientifically sound progress now that you've started here, okay? Microvasculature is an extensive subject of interest across our university. But coming specifically to your slides, firstly on your slide number 6,
this one?
That's right. Fiji is an open source scientific platform. I like Fiji. I can get into the bare bones of Fiji. However, the one on the right-hand side, the one with the feather in it, there's no way we can be supporting that. So okay. I'm strongly of the opinion that we stick to things such as MATLAB is the worst you should do. You should stick to Fiji and everything low level, okay? Because eventually, if this data was large data for you, then eventually this Procreate business is not going to work in your favor, okay? And secondly, if you go to your slide number 11 now there may be biological terms that say your experiment number 9 is an outlier or anything. But fortunately or unfortunately, you've involved yourself in something called precision something or the other. So I so very wish that we together, you and me and everyone else, we look into the reason why the number of bifurcations went up while you have number of bifurcations going down consistently in all your other observations, okay? Yeah. We have to get into experiment 9. I have not seen your outcome of experiment 9 yet before now. Okay. Thanks for that, Gabrielle. Very well done.
Hey, can I ask another question?
Yes, sir.
Sorry, this is just something for the future because I know Sanjay brought it up yesterday. Can you use your technique to identify a fractal dimension for the network? And have you tried to identify? So I guess that's a dumb question. First question is, can you identify a fractal pattern in your branching structure? And yes, if you haven't, that's okay.
I haven't looked into that yet. I haven't looked into that yet, no.
It's definitely possible. It's there.
Because yeah, determining the fractal dimension would be helpful with some of this as well, I think. But yeah, we'll have to talk more about this. There's a lot of things that there's a lot of moving parts here. So I think it's an excellent presentation, though. Really good.
Thank you. Are there any other questions? I can't see. Yusuf, did you have a question earlier? I saw your hand.
Gabrielle, I propose that in the interest of time, we move on.
Okay, you can ask me later, Yusuf. Thank you, everyone, for your questions.
Thank you, Gabrielle.